Why we stain bacteria. Hello viewers. Welcome to our YouTube channel. Today, we discuss about the gram staining procedure of bacterial culture. First, we clear the concept of history about that. Gram staining is first introduced by a Danish bacteriologist Hans Christian Gram in 1884. It's mainly work for the differentiate between gram-positive and gram-negative bacteria. Here, the some difference between gram-positive and gram-negative bacteria such as the gram-positive bacteria has smooth cell wall with a thickness of 20 to 80 nanometer. It has thick peptidoglycan layer to retain crystal violet stain. Then, the gram-negative, it has wavy cell wall and the thickness of cell wall is about 8 to 10 nanometer. It has thin layer of peptidoglycan. That's why it leaves CVI complex and retain counter stain. If we discuss about the morphology of these bacteria, then the gram-positive have a cocci, having 2 rings in a basal body, lipid content very low and having exotoxins. For example, Staphylococcus and Streptococcus. Instead the gram-negative bacteria morphology. It has non-spore forming rods, 4 rings in their basal body and having a lipid content of about 20 to 30%. It has both exotoxins and endotoxins. For example, Escherichia salmonella, so there are some difference. Now, we start the practical work. First of all, we have to preparing a broth solution in which we grow a bacterial culture. We need 1 gram of tryptin, 0.5 gram of yeast extract, 1 gram of sodium chloride and 100 milliliters of warm water. Mix the ingredients with the help of vortex mixture. Put the previously grow bacterial culture, both the petri dish and broth solution are placed in a laminar floor hood. It helps to protect the culture from dust and contamination. Then, we use the inoculation loop, place it on a flame until its tip become a red hot. Put a colony with this red hotted tip and dipped it in a freshly broth solution. Then the broth are placed in a shaking incubator for 24 hours. After 24 hours, the solution turned yellowish from translucent, means the bacteria can grow in this solution. This is called turbidity. Now, we have a freshly bacterial culture. The second main step is preparing a smear of that culture. Put a 1 drop of a culture on a slide with the help of dropper and spread it in a normal layer, not a more thick or more thin. Then, heat fix the drop as you seen in a video. Then apply a crystal violet stain on it and wait for the 2 minutes, then washed with a tap water. Second, apply with the iodine that forms a CVI complex and then washed with a tap water after 2 minutes. Third, apply an alcohol and wait for 30 seconds. After applying alcohol, the gram-positive retain CVI complex and gram-negative bacteria leave and again become colorless. Then, the last we apply a counter stain of safranin and the gram-negative bacteria retain and become pinkish in color. Dry the smear after the applying counter strain and observe under the 40x microscope. The bluish color of bacteria are gram-positive bacteria and the pinkish color are gram-negative bacteria. This is a simple gram staining procedure of bacterial culture. Subscribe the channel and stay tuned with us. Like the video and share with your friends. Thank you.